Hi, this is Miss Litton, and this is my wonderful bunch of AP biologists. Say hi.
Hi.
In here at our lunchtime. Some are eating sushi. It's delicious. Nummy sandwich. Okay, so we're talking about Chapter 13, Regulation of Gene Expression. We need to remember the process of using the DNA to make RNA is called transcription, and then using that RNA to build a protein is called translation. So we also said that the DNA is the same in every single cell, and so we have differential gene expression. So different genes are expressed in different cells. So we said, how could that be? And we talked about the different control mechanisms. So we talked about that you can control the process of what DNA is available to be transcribed. You can control the process of transcription, and that's the biggest point of control. You can control how it's processed, if it can leave the nucleus, how long it lives, if it can hook up with a ribosome, and then how that protein, once it's built, is processed. And then we investigated some of those different control mechanisms. So then we started off by learning the process with bacteria, and they're all about what kind of control, negative or positive? What are bacteria focused on? Negative control. Do they or do they not have a repressor? Now, is that repressor active? Is it inactive? That's a different thing. But if you're working with repressors, that's referring to negative control. Where do repressors bind? Yeah, remember, it binds to the operator. Where does RNA polymerase bind? To the promoter. So you want to remember your different areas and regions of the DNA and the role that they play. If you're going to have a repressor, a repressor is a protein. So there must be DNA that codes for that protein. What is the name of the DNA that codes for a repressor protein? Regulator, yeah. So your regulator gene that you see right here, if it is transcribed, meaning you make mRNA, and if that mRNA is translated, you are going to build a repressor. In this case, it's an active repressor, but you could have just as easily have built an inactive repressor, right? Either way, it codes for a repressor. The other key spots is the promoter. Who wants to bind to the promoter? RNA polymerase to initiate transcription. But it has a hard time binding if next to it the operator has a what on it? Repressor. So in essence, if you have an active repressor that is on the operator, you have shut down that entire operon. Remember, an operon is a group of prokaryotic genes that operate together. They work together for some end product that you're trying to make. An operon includes the promoter, the operator, and whatever genes you're controlling. The regulator gene is usually farther upstream and out of the operon itself. Okay, so then we talked about, we got this down, right? So I can jump ahead? I'm jumping ahead. Do you need me to do this? Do you need me to do this one? Are you good? Speak. Okay. Bueno? Okay, so then we talked about, let's look at this situation where a regulator gene is coding for a repressor, but this repressor is inactive. Okay, it is currently nothing, it's not working in any way, it's just sitting on the sideline, and so RNA polymerase is able to get on the what? Promoter. And if RNA polymerase is on the promoter, boom, 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 and you're going to make your mRNA, and that mRNA is going to be translated on a ribosome into some sort of protein. Okay? This is all about building some sort of protein. In this case, you're building 5 different proteins, 5 different enzymes who are going to work together to give you the end product of this amino acid, tryptophan. Okay, so let me walk you through that. If those structural genes are transcribed, the protein you will make from the translation of that transcription will be enzymes. Those enzymes will work together to build an amino acid, tryptophan. Okay, that's your end product. when I have enough tryptophan and I don't want to build any more tryptophan, the tryptophan acts as a co-repressor, activating our repressor. So now the repressor is on the operator, so RNA polymerase cannot get on the promoter. Is that a bad thing? No, it's a good thing, because I don't need any more tryptophan. But when my tryptophan gets used up, then there's no more tryptophan to act as a co-repressor, and RNA and the repressor will be off, and RNA polymerase gets right back on the promoter and starts transcribing those genes. This is called the TRP, right? Operon. It's an example. It's just one example of how genes can be regulated. It is negative control because it's using a repressor. This is called a repressible operon. Why? Because it's normally on, but it can be repressed. Normally on, but it can be repressed. Now, turn towards your bio buddy. Are you in the shadowlands of any aspect of that explanation? And let's get those questions answered. Go ahead, discuss. Okay, come back to me. Come back to me, guys. Be here so we can move on. Clarifying questions.
An operon is on if more tryptophan is needed, right? And off if not?
An operon is considered on, okay, if there is no repressor stopping it.
Okay.
An operon is considered off if there's a repressor in the way. Okay, what else? Yes?
So, I know you said, just to clarify, so like an operon can be said to be on for a repressible operon if, like, it's on, negative control means that it's on normal.
Okay, so negative control, when you use a word like negative control, all you're saying is, when you say the words negative control, is that a repressor is involved. That's what negative control means. Negative control is about a repressor, because repressors can put a stop to it. Very negative, put a stop to it. Okay? If I say it is a repressible operon, it is not normally repressed, but you can repress it. If I say it is an inducible operon, it's not currently being induced, but I could induce it. I can induce it by taking a repressor away. The functional word here is what's the repressor doing, because it's all about negative control. So in a repressible operon, your repressor is inactive, but I can make it active. Okay, more. Clarifying questions. Okay, so let's talk inducible operon. And this is the lac operon. In this case, the regulator gene codes for mRNA. That mRNA is translated into a repressor protein that is functional from the get-go. It's born ready. Okay? Because it is born ready, it will attach to the what? Who does the repressor always like to connect with? Operator. So the operator shut down. It's being it's got a repressor on it, but I can get it to leave. What can I get that repressor to leave with? With lactose. And lactose is considered what? Starts with a vowel. Inducer. Lactose is the inducer. So this is classified as an inducible operon. It is not currently on, but I can turn it on with an inducer. And that inducer, the lactose, when it binds to your repressor protein, it then removes it, because the repressor can either bind to the operator or it can bind, in this case, to the lactose. One or the other, but not both in this situation. So it removes the repressor, RNA polymerase gets on the promoter. We're going to transcribe these genes and translate them ultimately and form an enzyme that will digest what? Lactose. This is ultimately some way to regulate a catabolic reaction. Catabolic because we're going to break down what? Lactose. We're making the enzyme we need to break it down. I don't want to make that enzyme to break down lactose unless I have what present? Lactose. And then to put a little twist on it, I don't even want to make that unless there is no what? Glucose. And to ensure that RNA polymerase won't bind to that promoter unless there's RNA polymerase can have access to the promoter, but it won't bind unless there is a transcription factor, CAP, right, who is activated by the absence of glucose, right, in the absence of glucose. So that levels it up. I'm going to do the level up in just a minute. Check with your bio buddy, see if they understand an inducible operon. Go ahead.
So if lactose binds to
All right, clarifying questions. Clarifying questions you want to ask me? Okay, let's level up then. We know that E. coli would rather have glucose rather than lactose. So, when there is no glucose, okay, when there's no glucose, then you have an increase in cyclic AMP. Cyclic AMP, and we can remember CAMP, what word does that spell? And if you have a big camp, they're going to eat all the marshmallows. Yeah, all the marshmallows. Okay, all right. So, cyclic AMP is generated when glucose levels are low. And the beauty of that is now you have a chemical that will bind to a transcription factor. CAMP will bind to CAP. CAP will help RNA polymerase bind to the promoter to transcribe lactose digesting enzymes. And in that way, I can have a preference. If glucose gets me more from my bang, gets me more ATP, then if glucose is available, that's where I want to spend all my resources. Those are the enzymes I want to make. Okay? But if I don't have glucose, then I want lactose. So this is the way I can signal that. I can have cyclic AMP build up in the absence of glucose. It will turn on the transcription factor, and now RNA polymerase will bind to the promoter. What's the only reason RNA polymerase wouldn't be able to be assisted by CAP? What if RNA polymerase can't get on the promoter? If what? If there's a repressor on the operator. And why would there be a repressor on the operator? Because there is no lactose to digest. So this will ensure that I will not make lactose digesting enzymes unless A, lactose is available to get rid of the repressor and glucose is absent, so I get help to get RNA polymerase on the promoter. Yeah? All right. So real quick, I'm not even going to have you click in on these because I want to get to some other items. Just take a look. I'm going to, when I point out, then you're going to give me the answer. Tell me the words you learned. What's the word? Promoter. Good. Just read. Just read. Tell. Perfect. Read. Tell. Yeah, regulator. Okay? Regulator.
Oh, yeah.
Regulator codes for the repressor. Read. Tell. Lactose. And lactose acts as a what? Inducer. Good. All right. Read. Tell. Structural genes. Good. All right. Then we said this is assuming we can even do transcription, that the operator is available, regulator, all of that. This is assuming we can even do that if the DNA is undoing its what? What do we call that? Supercoiling. Is the DNA wound up or not wound up? And so what happens is you can attach little methyl groups to the DNA, you can attach little methyl groups to the histone, and if it's attached to the histone, it makes it even relax even more. If it's attached to the DNA, it coils it up tightly. So depending on if you attach it, and that's all, I can't even access those genes that code for proteins, right? So this is above whatever's in there. I don't know if that's a dominant gene that's supercoiled or a recessive gene that's supercoiled. I'm not expressing it, not because it's a recessive trait. I'm not expressing it because it's supercoiled. So we call that what kind of genetics? Epigenetics. Epigenetic inheritance. Anything that's going to influence whether you can even read those genes and transcribe them is epigenetic inheritance. And we looked at examples of that. Well, first of all, we could visually see it, whether it's heterochromatin or euchromatin. Heterochromatin is darkly coiled DNA that's inaccessible, lightly colored here in the euchromatin, okay? And we looked at bar bodies, and that second X stays condensed, and you don't know in each cell which X is going to condense. So in one trait, if you have the orange one and the black one here, in some cells the orange X would be expressed, and in some the black X would be expressed. Okay? And that creates a mosaic. So that's one form of control. Transcriptional control, we've just spent time going over transcriptional control. But we went over transcriptional control in a prokaryote. In a eukaryote, it's not so much about negative control, it's more about the positive. And the transcription factor, the cap that was activated by cyclic AMP, that's a form of positive control. And in eukaryotic cells, it's more, do you have this and this and this and this all facilitating RNA polymerase to be transcribed? Okay, so all of these things have to be activated. We added another region onto the DNA, the enhancer region, okay, where an activator can bind to get a transcription factor ready to then help RNA polymerase bind to the promoter. And in this way, since we don't have operon, we could have on chromosome 7 something we need, chromosome 3, chromosome 27 we need. But if they all get flipped with the same transcription factors, as long as I have those prevalent, right, then I can transcribe and translate those genes as collectively, as a unit, even though they're located on different chromosomes, because I have the same chemicals available to me in that nucleus. Do you follow that? So they can be regulated as a unit, even though they're not next to each other, because they have the same switch. And we said it can be very detailed, all the things that you have to have in order to get that to work. And we talked about, I mean, it's not like this is an AP level question here, okay? So that the transcription factors, you call it positive, it's more about the yes, it's not about the no. It's more like a Boy Scout, right, than a sniper. I would say a sniper is more like negative control, right? Either the sniper has bullets or he doesn't have bullets. Either the repressor is functional or it's not functional. Okay? So we have talked about, is the DNA available? Is the chromatin, is that DNA is available? Can we transcribe it? Can we boom it? But once we boom it, we know we have to put on a cap and a tail and cut out the introns before it leaves the nucleus. So that is called post-transcriptional control. Post-transcriptional. I made the mRNA, I boomed it, but I have to do this part to it. Okay? Then once it leaves that, then there's other aspects of control. Now, the big thing on post-transcription is what introns are removed. So I can have this one book, right? But depending on what words I cross out, I could write different poems with the words in this book. Even though it would read, right, left to right in the original, it would read like an AP Bio, I could actually write poetry by crossing out all the words I don't want, right? Couldn't I? I just have to cross out the right words. So that's what I'm saying. You can get many different poems from this one book just depending on what words I cross out. Okay? So it's combinations of those words. Are you getting that? Okay? So depending on what introns I cut out depends on the ultimate protein I get. So I can use the same, when we say this gene codes for this, Well, this gene also codes for this, and this gene also codes for this, depending on what introns you cut out. Doesn't mean there's just one kind of protein that it codes for. Okay? Then we said that was post-transcriptional control, and then we talked about translational control. How long does that poem last, right? What's the life expectancy of it? Could it get digested? Could another single-stranded or small interfering RNA, siRNA, get there and bind to it, and so that it can't be translated? Those are some of the other concerns that you would have. Does it get further modified? MicroRNAs destroying it. How long? Can it hook up with the small ribosomal subunit? Can the large ribosomal subunit get hooked up? That's all translational control. And then post-translational control, let's say I make that protein. It has to be folded in its correct shape in order to be functional. Some of the amino acids might actually have to be cut out of it before it's functional. Okay? Okay. Do you need me? Okay. So, how long does that protein last?
But it'll happen in a minute. So.
What? What did he say?
It'll happen in a minute. What's going to happen on the faint, but I won't disturb you now.
Okay. So how long that protein lasts. Okay? Now, we know, big ticket, okay? We know the DNA codes for the RNA and the RNA codes for the protein. So where are levels of control? Is the DNA even available? Now once it's available, is anybody getting in the way or helping the RNA on there? Once I've made the RNA, how is it getting processed? Can it get out of the nucleus? Can it get hooked up to a small ribosomal subunit, to a large ribosomal subunit? The protein product I make, do I need to fix or adjust it? Do you see all the levels of control? So now, what happens if this whole process and my DNA is screwed up? This is assuming that my DNA is okay. Now, what if my DNA is broken? What if in the process of making my RNA I screw up? These are called mutations, okay? And it's called a point mutation if you can point to a single nucleotide that's causing it. Now, when we studied our genetic code book, right, our codons, we could look up, oh, this series of 3 makes this codes for this amino acid, right? So now we saw sometimes there was more than one way to get the exact same amino acid. So that's why when you have this wrong DNA, which gave you the wrong RNA, this is called a what kind of mutation? Silent. Because even though it's wrong, it still gets you the same end result of the same amino acid. So it's a mutation, but no biggie. It's all right. We got the same result. Okay? On this one right here, you were right in the middle of the sentence, and every time you fall for it. And this is called the stop codon because you're not completing your chain of amino acids because you coded for one of the 3 stop codons. So this was protein interruptus, okay, right in the middle of it. Then here, here we have put in the wrong amino acid. So as a result, wrong amino acid, wrong folding pattern, wrong shape of the protein, protein is non-functional. Okay, so that's one way it can go wrong. Then one of the worst kind of mutations were frame shift mutations. And when we looked at that, I gave you 3-letter words to represent 3-letter codons. Okay? And in this case, here is our first codon is THE, but we've removed the E. So now our first, hello, my studious children. My first codon instead of THE is TH what? B. Okay? So then my next codon is IGF. And every one of the codons, this is not a point mutation, this is a frame shift because I'm off. I'm reading in the wrong frame now for all of them. So every flipping amino acid is probably going to be wrong. So that is a real problem, okay? And then I gave you some examples of non-functional proteins. They're phenylalanine. Okay, phenylalanine, you need this enzyme right here. If it's faulty, you have PKU because you have a buildup of phenylalanine. And this one, you need tyrosine. And if you don't have this enzyme functional, you'll be an albino because you don't lay down any melanin. This is all coming from, this has got to be changed into this, has got to be changed into this. So if it's screwed up at the beginning, then you're all of these things. And the example here was, this is a female, but her Y chromosome is not getting transcribed and this is a male, but her Y chromosome is not getting transcribed and translated. So she is a girl. Okay? Because her Y chromosome is in essence silent. Okay, and then I had some clicker questions on here. So what kind of control is it where the mRNA is getting introns removed? That's post-transcriptional. Bar bodies is chromatin structure. Yeah. You're welcome, sweetheart. Ability to bind to the ribosome. Translational. Folding pattern. Post-translational. And then I've already talked to you about cancer. We've learned about those genes. And that's the end. Yay! Good job. I'm super proud of you. And if you're up really late, eat some toast.